hi, very happy good evening. So this is Krishna Ma'am. We'll be talking about the story of life on earth, and that's biology. So thank you so much for joining my new beginning series. So today we're going to talk about photosynthesis in higher plants. So photosynthesis is a very important chapter when you talk about plant physiology. So we're already done with one chapter in plant physiology, that is transport in plants. Now we are in the second topic, so actually not second, so in our order it is second. So we're going to talk about photosynthesis in higher plants. So when you talk about plant physiology, two topics are of utmost importance and they give us a lot of base understanding. So what are those topics? First one is photosynthesis in higher plants. The next one is respiration in plants. So we'll do these two chapters continuously, so there's a link and you understand the link behind this. Okay, so first we'll start off with photosynthesis. So probably it's a very important unit, so we might take 4-5 lectures, and then we'll go to respiration. Again, respiration is very important, we'll need 4-5 lectures. Okay, so let's finish off these two chapters continuously and then we'll move to class 12, or we'll probably move to biomolecules. All right, fine. So today we're not going to see a lot about photosynthesis, so you know our class is only for 20 to 25 minutes. So I don't know if anyone would take class for 20 to 25 minutes, but it's pretty chilled, right? So even when you want a recorded lecture, so you don't have to look for a lot of time to fit yourself to watch the recorded lecture. You can easily watch because it's of a short duration. And yeah, I split up the topics so that you can easily capture it later. So today we'll talk only about the experiments that is involved in photosynthesis, and let's see how it goes forward. All right, so let me know if you guys need some more motivational session or something extra, so please do let me know. Okay, so here we go. So photosynthesis in higher plants, this is lecture 1 or part 1 we can say. So here we move forward. So what are the topics that we're going to discuss in this chapter, at least for today? So we're going to introduce, so I guess you all know what is the definition of photosynthesis. So why plants use photosynthesis, why plants do photosynthesis. So how do we benefit from photosynthesis? I guess you know all these base questions. So what do we know about photosynthesis? Yes, today you and I know what is photosynthesis. It's a process by which plants prepare food. But how do plants do? So what do we know so much about photosynthesis? So we know that sunlight is needed. We know that carbon dioxide is needed. So we know that it gives glucose and oxygen. How? So how did scientists find out? How did they do all that? Yes, Nag Pooja, very happy good evening. So thank you so much for joining my lecture today. So please do like, share and subscribe. So why didn't you turn up for my mentee on Monday? I was asking W about you. Alright, so we're going to talk about a series of early experiments who gave us a base understanding to photosynthesis. So these experiments gave us a very basic insight about what today we know about photosynthesis. So where does photosynthesis exactly take place? And I guess even a 6th grader would know, that is in chloroplast. So how many pigments are involved in photosynthesis? So you have 4 pigments and we are going to look into the 4 pigments. So what is a light reaction? So what exactly happens? What are the products of the light reaction? Where exactly does the light reaction occur? So let's see. So we'll talk about the experiments and where photosynthesis takes place and how many pigments are there. Okay. Yes. You want to consult options? Yes. Okay. So you want me to give you options on what? Science with Singania, yes, a very happy good evening. So thank you so much for joining my session today. So please do like, share and subscribe. So which option, like MBBS, nursing, homeopathy, etc. So that's already done. So if you would subscribe to my channel, and if you go to the videos, I've already discussed the options right after the NEET exam. So if you could rewind and see, yes, you get all the videos. There is no video that I haven't done, I would say. So yes, I have discussed options. I have given enough motivation. So if you want to look for options as to whether homeopathy is good, whether Ayurvedic is good, whether nursing is good, whether B.Pharm, M.Pharm is good, yes, already the videos on that has been done. Okay, so you can go. Acha, you went to consult. You went to consult eye doctor, isliye you haven't come. Alright, that's completely fine. So guys, if you want to weigh the options as to which one you want to prefer, Ayurveda, homeopathy, so please look into the videos. So it's already there in my channel. So if you subscribe my channel, you can go to the view videos and you can see all the videos out there. So let's see how much we can do today. So why study about photosynthesis? What difference would it make in our daily life? So animals, human beings, all of us depend on plant for food, right? Even if you are a very, very religious meat eater, probably you eat meat every single day, so still you depend on plants. How? So for you to have chicken in your plate, the chicken has to be fed, right? The chicken has to be fed with grains, and the grains come from plants. If you like mutton, the goat has to be fed and the goat feeds on plants, right? So directly or indirectly, all of us at one point or the other is dependent on plant for food, right? So plant is the major producer, I would say. So but where do the plants get their food from? So plants get their food from their own process, that is by photosynthesis. So green plants synthesize their own food through the process of photosynthesis. Thus plants are autotrophs. The one which can prepare their own food, the one which can prepare the self food, that is autotrophs. Ma'am, aren't we autotrophs? I also make my own food in the kitchen. Right, so you and I are not autotrophs, we are still heterotrophs, because the raw materials also we get from plant, right? So at the end, if you have potatoes and say if you have egg, everything comes from the one or the other, right? So the raw materials, you are dependent on plant. Say for example, you have carrot and radish and you're going to make some very nice sabji. So again, you're dependent on plant. For all the spices, where does chili come from? Right, so you are directly dependent on plants, so you cannot call yourself as autotroph. You can call yourself as independent cook. So, but yeah, plants are the autotrophs. So photosynthesis is a physiochemical process by which it uses light energy to drive the synthesis of organic compounds, right? So this is how we define photosynthesis, as the physiochemical process, okay, by which it uses light energy to drive the synthesis of organic compounds. The organic compound here would be glucose, because the end product of photosynthesis is glucose, right? All living forms depend on sunlight, so that is the main source of energy. So for plants to prepare food also, they need sunlight, right? So sunlight is the main source. So for all animals and for all humans, plants are the ultimate source of food. And for plants to make food, sunlight is very, very essential. Thus sunlight is very, very inevitable for all the living organisms on earth. That is the conclusion. Alright, yes. So here we move. The use of sunlight by the plants for photosynthesis is the basis of earth. So that is how the life moves on in earth. So release of oxygen into photosynthesis by plants. The end product of photosynthesis is that your glucose and oxygen molecule is produced, and the oxygen is given out into the atmosphere, because of which you and I breathe, right? So this is about your photosynthesis. So this chapter we are going to talk about the structure of photosynthetic machinery, various reactions that transform light, chemical energy. So this is your photosynthesis. But as such, this is very interesting because it gives you a lot of new information which you and I would have never thought of even in our wildest dreams. So what do we know so far? So we know some basic things, like for the plant to perform photosynthesis, they have to have chlorophyll molecule, right? And yeah, they need light, they need sunlight, they need carbon dioxide. So these are required for the process of photosynthesis. So this much we already know, right? So first, I think you would have performed this experiment in your lower classes. Even if not, you can do it at home also. So to look for the starch formation in two leaves, okay? So you take a leaf like this, okay? So at one point in the leaf, you put a black paper and let it like that and leave it undisturbed for some time. After 1 hour or so, add drops of a solution to check for the presence of iodine. Okay, so if the green surface of the leaf turns blue, means it has iodine. If iodine is present, it means it has starch. Ma'am, if iodine is present, how can you say it has starch? Because starch has a helical structure. Oh my god, acha. So starch has a helical structure like this, and in between these you have the iodine molecule stored. Ma'am, why are we testing for the presence of starch? The end product of photosynthesis is glucose. Starch is the stored form of energy. So glucose is what is produced at the end of photosynthesis. But this glucose is stored as starch in plants, and it is stored as glycogen in animals. Fine? Yes. So when you take a leaf and when you cover a portion of your leaf, right, for some time, and then after 1 hour, if you test for the presence of starch or iodine, so the portion where you covered with black paper will not give you a blue color, indicating that starch is absent there and photosynthesis did not happen because sunlight did not fall on this piece of area. Thus concluding that photosynthesis occurs in green plant only in the presence of sunlight. So this is a simple experiment which you would have done in your lower classes. The second experiment, so probably you would have done this again. So you have a plant. So if you see the entire plant is placed in sunlight, right? But you take one portion of the leaf and put it inside a glass jar. So now sunlight can fall on the glass jar. So I am closing the glass jar tightly, and at the bottom of the glass jar I have a KOH solution, potassium hydroxide solution. So what I do is I allow it through photosynthesis for 1 hour or 1.5 hours. Then I take this leaf and test for the presence of starch. Surprisingly, it does not test positive for the presence of starch. Though sunlight falls on this leaf, carbon dioxide is not able to enter this leaf. It is blocked here because I have screwed or corked the bottle. Sunlight falls, but without carbon dioxide, again plants cannot photosynthesize. So thus carbon dioxide is also equally important for the process of photosynthesis. Alright, yes, so these are two simple experiments. So apart from this, now we have a series of experiments that suggest the following. So series of simple experiments gradually led to the understanding of photosynthesis. So experiment 1, so you had scientist Joseph Priestley. So he was the first scientist. So Joseph Priestley also discovered oxygen. Alright, in 1770 he did a series of experiments in which the essential role of air in green plants was relieved. Okay, so he discovered oxygen for us. So what did he exactly do? So he took a jar, and inside the jar he placed a mouse and a candle. Okay, so he took a jar, he placed a mouse and a candle. After some time, the mice died and the candle stopped burning. So when he placed a plant inside the jar along with the mice and the candle, the mice continued to live and the candle continued to burn. So he gave a first conclusion saying, so first here in this case he gave a conclusion saying the burning candle and the animal breathe air, but they also damage the air, right? So that is why when you are in a closed space, we say you are not supposed to be in a closed space for a long time, please come out, because you are damaging the air. The same thing happened here. So when he put a plant, they continue to live. So he hypothesized saying that plants restore to air whatever breathing animals and the burning candle does. So your burning candle and your living animal damages the air, and that damage is restored by plants. So that is what Joseph Priestley's conclusion was. Okay, so experiment 2A. So why have you named 2A? Because the same scientist did another experiment which I have numbered as 2B. Alright, so here we have a scientist, he is called as Jan Eigenhaus. Okay, so similar setup was done by him also, just like Priestley, but what he did was he took the setup. So what did Joseph Priestley do? He had the setup, he had the mice, he had the candle, and he had the plants. Now he placed this entire glass jar in light and in dark. So in dark, when he placed, the plant did not grow, nor neither the candle was burning, right? Now when he placed it in sunlight, he found that yes, everything is fine, right? So he said that though plants restore the damage that is done by the burning candle and the animal, it is done only in the presence of sunlight. So plants take in carbon dioxide for photosynthesis only in the presence of sunlight, right? So only when you give light, so yeah, the plants also will work. So that was his first experiment. So sunlight is essential to plant process, the purify air fouled by the burning candle and the breathing animal. The second part of the experiment that Jan Eigenhaus did was an elegant experiment which set the aquatic plants to bright light. So he brought aquatic plants to bright light. When he got them to bright light, he found small bubbles that was visible. The same bubbles are not visible when kept in dark. So which means these bubbles are oxygen. So oxygen is released only in the presence of sunlight. Obviously, so photosynthesis happens in the presence of sunlight, and as a byproduct oxygen comes out, and with the evidence of the bubbles he found out that yes, green parts of the plant releases oxygen. Not all parts of the plant releases oxygen, only the green part of the plant releases oxygen. Right? So next we have a third experiment by a scientist who is called as Julius von Sachs. So Julius von Sachs was also the scientist who came up with the concept of hydroponics in mineral nutrition. So hydroponics is a condition where you grow the plants in the absence of soil but in a defined nutrient medium along with aeration and water. Alright. So here Julius von Sachs gave us an understanding that yes, when plants grow, glucose is being produced, which means as plants grow, they photosynthesize and produce glucose, and this glucose is stored as starch. So the final conclusion of his experiment was that there is a green substance in plants, which today we know it as chlorophyll in plants. So they are stored in special bodies within the plants, which we today know as chloroplast. So the chlorophyll present within the chloroplast is responsible also for the formation of photosynthesis. So conclusion, glucose is found in the green parts of the plant and it is stored as starch. This was the conclusion that Julius von Sachs gave. Understood? So then you have experiment 4. So that is done by the scientist T.W. Englemann. Alright. So what did he do? So he split a prism of colors. So when I join, a prism has 7 colors, right? The colors of the rainbow. So when I join all the 7 colors, I get white light. Now when I split the prism, I get the spectral components of all the colors, right? So now into the spectral components, so I have split the prism here, and into this mixture, I put a group of green algae, Cladophora. Okay. So what will green algae do? It will align itself along the wavelength of photosynthesis because green algae has the ability to photosynthesize, right? So they will align along with the wavelength of photosynthesis and they will start doing photosynthesis. But how do I confirm photosynthesis has happened or not? So I introduced, or T.W. Englemann introduced, a group of aerobic suspension bacteria. So aerobic bacteria will need oxygen. So oxygen is released as a process of photosynthesis. So the bacteria will move towards the place where oxygen is available. So in a way I can find out where photosynthesis is happening. Yes. So he split a spectrum of colors. He split a prism of colors, and into that he put a group of green algae, Cladophora, and now he also put a group or suspension of aerobic bacteria. So the green algae would align along the wavelength of chlorophyll to do photosynthesis, and as a result of photosynthesis, oxygen comes out. The aerobic bacteria which are in need of oxygen will come closer to the region where photosynthesis is happening, right? Thus the first absorption spectra of photosynthesis was verified, was observed here, you could say. So this is what T.W. Englemann did, right? So the first action spectra of photosynthesis was thus described. So the action spectra of chlorophyll was very close to the absorption spectra of chlorophyll A and chlorophyll A. So that is where the wavelength that they can observe light, right? So one last experiment. So by the middle of the 19th century, so Joseph Priestley is working since the 18th century. So by the middle of the 19th century, the key features of photosynthesis were clearly known. So plants to photosynthesize need carbon dioxide, they need sunlight. The green plants are the ones which produce oxygen, the green parts, right? And glucose is stored as starch, and glucose is the byproduct of photosynthesis. So empirical equation of the entire process of photosynthesis is carbon dioxide plus water in the presence of light gives glucose plus oxygen. So CH2O is the generalization of carbohydrate. So this is how the empirical equation is. So the last experiment in series is done by Cornelis van Niel. So this was a milestone contribution. So this gave us a lot of understanding in future. So what was this experiment? So here we go. So he said that the oxygen that is released as a product of photosynthesis comes from water and not from carbon dioxide. So we thought the carbon dioxide, so the oxygen that comes from carbon dioxide. But Coronius 1 Neal said that the oxygen that is produced during photosynthesis comes from water and not from carbon dioxide. So he studied the purple-green bacteria. If you remember, they are the halophiles, I would say. So these halophiles, what they did, they also do photosynthesis. There are two types of autotrophs, if you know. So one is chemoautotroph, one is photoautotroph. Photoautotrophs prepare their food in the presence of light. Chemoautotrophs prepare their food in the presence of chemicals. Alright. So these purple bacteria, they prepare their food in the presence of chemicals. Okay. So when they prepare food in the presence of chemicals, oxygen is not released. Okay. So if you look here, so this is a normal process, right? So you have H2A because oxygen comes from water. So here the bacteria which prepares food, which are chemoautotrophs, do not utilize water, right? So they utilize chemicals, H2S, hydrogen sulfide. So when it is H2S, only S is evolved, not oxygen is evolved. So that is why not all photosynthesis is beneficial to us. Whichever photosynthesis can give us oxygen, only that is beneficial, right? For example, imagine a chemoautotroph is using H2S plus carbon dioxide to get glucose plus sulfur. So if it's water, then you have carbon dioxide. So sorry, you have oxygen. So the oxygen comes from water and not from carbon dioxide. So this is the conclusion Coronius 1 Neal gave us. All right. Yes. So green plant in water, in green plants, water is the hydrogen donor and it is oxidized to oxygen. All right. So your water is the one that is getting converted to oxygen. Some organisms do not release oxygen because they do not use water. Right. So in case of your purple sulfur bacteria, H2S is used and sulfur is the end product or sulfate is the end product. Alright. So conclusion is oxygen is evolved by the green plant, comes from water and not from carbon dioxide. So this was later proved, or the evidence was given by radioisotope technique as well. So this molecule of oxygen comes from water molecule. So this is the final equation of photosynthesis. Alright, yes. So we have done with the major part, I would say, because the experiments are very, very important. So I want you to remember the experiments. It's okay if you don't remember the time or tenure of the scientist, but yeah, the experiment, the conclusion, the name of the scientist is absolutely important. Alright, so we'll stop here and next class we'll talk about what is a Z scheme. So what is for PS1 and PS2. Alright, so I'll meet you on Friday at 4:30. So until then, this is me signing off. So have a great day, great evening, and I'll meet you on Friday. Okay, so thank you and bye.